This is joint work with my students, Mohsen Zakari, Avi Shrivastava, and Fatem Al-Madarosi. Mohsen was supposed to come give this talk, but he ran into some visa issues at the last moment. So if you see anything here that you find interesting or that you like, I highly encourage you to tweet and tag him, make him feel a little bit better about not being able to make ISMB this year. So I'm going to talk about transcript quantification, and as the last speaker pointed out, it's a computationally difficult problem, but it's been studied for a while, and there are some reasonably good solutions for it. So let me give you an overview of the problem. So we have an RNA-Seq sample, and this is what you see over here, and we're conceptually thinking of it as some mixture of transcripts. And the reason for this is that transcripts are the unit of expression in the cell, and that's the thing that we really want to determine the relative abundances of. But each transcript comes from a gene, and a gene can express multiple isoforms simultaneously. So here we have M genes, and each one is displaying some number of isoforms in some relative frequency. And then we have a measurement process, which for us is the RNA-Seq assay. And there are many different protocols, but it produces essentially tens or hundreds of millions of short reads. And so we don't see things this short anymore, but it's common to see, 75, 100, 150 base paired end reads. And now what we need to do is to go backwards from this measurement, and we want to infer the relative abundances of the different genes, and the relative abundances of the different isoforms, right? So basically we want to recapitulate the mixture percentages in our original sample from these measurements, and in our case, knowledge of what the transcript set is. So we can think about this as stating the problem this way. Given a collection of RNA-Seq fragments and a set of known or assembled transcripts, we want to estimate the relative abundance of each transcript. And so when we think about this, we like to think about what is the mathematical model underlying it, and how do we model the thing we're actually trying to measure. And so the particular quantity of interest for us is going to be this thing that we call the nucleotide fractions. So we denote them as eta. And really this is just saying that in an unbiased experiment, sampling fragments from a particular transcript type, of here there are 3 species of transcripts, blue, green, and red. It depends on 2 things: the number of copies of that transcript present in the sample and the length of those transcripts. And the product of those 2 things determines the fraction of nucleotides in your sample that are sequenceable from that fragment type, from that transcript type. And so you end up in this sample, for example, with about 30% blue nucleotides, 60% green nucleotides, and 10% red nucleotides. And so the way that this was originally posed by Bowley and Colin-Dewey in the ISMB paper was to think about the generative process of RNA-Seq as a 2-step process, and this is a simplified model. Well, what we're going to do is first we're going to choose a transcript from this experimental mixture to generate a fragment from, and we're going to pick this transcript proportional to the total available nucleotides, which, as I just said, is the count times the length. And then we're going to pick a position P on this transcript uniformly at random to sequence. So if we have this sequencing oracle that produces reads in an unbiased fashion, we expect to see something like this. We see a couple of green reads, some blue reads, and if we sequence deeply enough, we eventually see a red read. It might take a while because the abundance is pretty low here. And we keep going until we have this mixture, right? And so this is just another way, another cartoon showing that we want to go backwards from this and infer this. And so this would be easy if every fragment had a unique transcript of origin, if we could assign every fragment uniquely to the transcript that it comes from, but because of alternative splicing and sequence homology, this is not generally possible. And for example, in human RNA-Seq experiments, we might see something like 80% multi-mapping at the transcript level, so it's quite a big problem. And so underlying this generative process is actually a probability function. And so what I'm writing out here is the probability function underlying RNA-Seq quantification. Again, this was originally proposed by Bowley and Colin-Dewey in the ISMB paper. And so what they say is that the probability of observing a given set of fragments depends on your estimated nucleotide fractions and your known transcriptome. And the first assumption that's made is that fragments are sampled independently. So the probability of observing my entire data set is just the product of the probabilities of observing each of my fragments. And then we can further expand this so that we have a product over all of our fragments and then a sum of this 2-step process. First, what is the probability we chose a particular transcript TJ? Next, what is the probability of generating a particular fragment FJ, TI, given that it came from this transcript TI? And so this inner sum, even though it's technically over all transcripts, is often approximated by only the transcripts to which that fragment aligns. So if a fragment doesn't align to a transcript, this whole probability becomes 0. And so we want to find the values of eta that maximize this, so we want to turn this probability into a likelihood. And luckily we can do this at least locally using an EM algorithm. And so that's great, but what is the problem? So, if you've been following the literature, there's been a lot of work since RSEM, even though it's sort of a great model and a great program, on transcript quantification. And one of the problems is that this is a slow process, and one of the reasons that this is a slow process is if you look at this EM algorithm, we have a product over fragments and a sum over all the mappings of those fragments. And so typically, we have on the order of 10 to the 7th to 10 to the 8th alignments in a typical sample. And we have on the order of hundreds to thousands of iterations for this EM procedure to converge. And so this means that if each iteration of the EM scales in this number, and we have this many iterations to do, it typically takes a long time to actually perform this optimization. And so there's a trick that has been commonly used to speed this up considerably. And so this is to partition fragments into equivalence classes and to treat groups of fragments that map in the same way as one unit in the inference process, to treat them as sufficient statistics of inference. And so this equivalence classes are defined by this equivalence relation. It says two fragments F and F prime are equivalent if and only if they map to exactly the same set of transcripts. So here's a cartoon example. I have an experiment with 4 fragments and transcripts A through F. But transcripts 1 and 2 map to the exact same set of transcripts. They map to B and E, while transcripts 2 and 4 map to transcript C. And so even though I have 4 different fragments in my experiment, I only have 2 types or classes of fragments. And so we can think about representing this as a table where we label the transcripts, the fragments by the transcript they match to, and then we have the count of each. And so this idea, as I said, has been around for a long time. There are multiple different formulations. They're not all equivalent. But the one that I'm talking about here was first introduced in 2011 in this paper by Ernest Turo with a program called MMSeq. But this idea spans the approaches based on traditional alignment and then sort of the new lightweight quantification approaches that are alignment free. So the idea of factorizing the likelihood is independent of how you generate the mappings. And so why would we want to do this? Well, the reason is you can sort of see empirically, so theoretically it makes sense, it's intuitive, but empirically the number of these equivalence classes is often 2 orders of magnitude or more fewer than the number of alignments. And so if I have an EM algorithm that scales in this number, it's automatically 2 orders of magnitude or more faster, right? So that's really the motivation here. But it's not for free, we're losing something, right? So what are we losing? So the original likelihood allows to have a specific conditional probability for every fragment transcript pair that you see. But in the factorized likelihood, we're saying that within an equivalence class, all fragments have to have a specific conditional probability, one scalar, with respect to a transcript in the class's label, right? So we're basically getting rid of or completely collapsing this term. And so this is really what our paper is about. We're asking, how much does this matter? Does it make a difference? And can we improve on it if it does? And so why might it make a difference? What's the intuition for this? Well, consider one thing that goes into this conditional probability, which is the probability of observing a fragment with the given insert size. So I have a single fragment here that multimaps to 2 different isoforms of the same gene. And the sequence alignment scores are identical because it maps to identical sequence, but the implied insert size is drastically different. And so if I know that my fragment length distribution in my experiment has a mean of approximately 200, then this mapping or alignment is a priori much more likely than this one. All right, and so if we throw that information away, we might be giving something up. And so we tried to answer this question first via simple simulation. So what we did is we took a multi-transcript gene here, RAD51, and we took 10 transcripts and we simulated reads coming from them drawn from this uniform distribution, and we did this with a polyester simulator, and we did this experiment 30 times over so we could get a distribution of essentially relative differences. So here lower is better. And here what I'm showing is different tools, right? So we have our sort of fast tool that's based on an approximate factorization called Salmon. We have RSEM, which uses the full model, a variant of our tool which we took the factorization away from, and then this version so Salmon is sort of a little bit intelligent here in that it averages the conditional probabilities over an equivalence class. But some other tools and some other ways of doing it just say, forget this altogether, make it a uniform conditional probability prior. And so what you can see is having the conditional probability here for certain transcripts does seem to help. And the difference is magnified as you increase the coverage, right? So as you have more coverage, you're getting more and more different types of fragments and more useful information from these conditional probabilities. And so it does seem that potentially we're losing something here. So we're interested in recovering it. So how might we do that? So we proposed a couple things in the paper. We tried a few things that don't work, but this is the one that did, so it's the one I'm going to tell you about. So recall the original definition of equivalence classes. Two fragments, f and f prime, are equivalent if and only if they map to the same set of transcripts. So what we're going to do is we're going to define a function which takes a fragment and a set of transcripts. And it's going to return, and it's dependent on some parameter k. So this can be set, but we have a way of automatically setting it. What it returns is a conditional probability bin for fragment f with respect to each of these transcripts. So basically, we're discretizing the space of conditional probabilities, and we're saying, what bin do I fall into for each transcript in terms of my conditional probability of generating this fragment from that transcript? And we define a new equivalence relation. So we say that two fragments are equivalent if and only if they map to precisely the same set of transcripts, and they have the same conditional probability vector, right? So here the vector is over integers, it's over indices of bins, and so it's very easy to sort of check this relation. And so you can think about this intuitively as well. So what I'm showing here is an equivalence class with only two transcripts, T1 and T2. And here, what I'm drawing on the x-axis is the conditional probability of seeing a particular fragment given T1, and on the y-axis, the probability given T2. And so these sum to 1, because these are our normalized conditional probabilities. And furthermore, you see everything sort of along this diagonal for that reason. And so the original approach of just sort of replacing these conditional probabilities with a single aggregate, if you averaged over all of these fragments, so there's 60 fragments here in this picture, each one a black dot, you would end up with something here, right? So it's essentially the same as the uniform model. And so in our model, what you get is you get replacing that with these local averages, right? Because our equivalence relation is somewhat different. And so the thing that you really get is that the maximum error between a fragment's true conditional probabilities and what you're approximating it with is much smaller when you partition the space. It's sort of an obvious result, but you can tune that parameter to control the error. And so does this work? And how well does this work? So this is what we tested out. So here I'm showing you the same plot that I had before, except I've added a few other tools. And so again, the red is the sort of completely unweighted equivalence class representation. The blue is the normal sort of pre-existing factorization that does an averaging. And so again, you can see that does buy you something. But this factorization I just told you about, we call SAM and RF for range factorization, performs essentially indistinguishably from the full model, the purple, and from RSEM, which for us was the reference implementation. All right, so at least in this small scale example, you see that it recovers the differences in modeling accuracy that we'd previously lost. So that's a very sort of simple one-gene experiment, and it's just a proof of concept. And so how does this change things on the transcriptome scale? So we used another simulator. We used RSEM-Sim, and we did a transcriptome-wide simulation. And so here what we did was we learned parameters from real data, and then we simulated data, so we know the ground truth, and then we measured two error metrics. So the mean of the absolute relative differences, so here lower is better, and the Spearman correlation, so here higher is better. And so again, RSEM is sort of our reference, and what we can see is there are some small differences. The MARD is lower in our range factorization, and the correlation is higher. It's not a huge difference, but it does recover what we sort of saw with RSEM. And so we were interested in why this might not be so huge, at least given what we saw in our small-scale sample. And one potential reason is there are tens of thousands of expressed transcripts in this experiment. And so many of them are very easy. Many genes are producing what is quite clearly only a single isoform. And so all the methods are doing well, and you're sort of washing out differences in this large sea of different transcripts. And so what we did is we restricted the set that we did this analysis on to what we call critical transcripts. So these are meant to be ones that are neither too hard nor too easy. So we classify these by, they have an absolute relative difference with respect to RSEM between 0.25 and 0.75. So they're sort of middle of the road in terms of difficulty of predicting. And so in this set, what we see is that the differences are considerably larger. And again, we see that this improved factorization essentially recapitulates the result of the full model, right? So we were able to recover what it was that we lost. We did experiments on real data as well, and here we don't have ground truth, so we treated RSEM as ground truth. And so what you're seeing here is distributions of Spearman correlations. Higher is better, means it's closer to RSEM, and the full model and range factorization of our approach are overlapping. You can't even see the difference, right? So at least within measurement error, we're sort of not finding statistically significant differences. And so it's still very efficient. So how does this change the number of equivalence classes? Because we're chopping these things up. So this is what we see in practice, that the number of equivalence classes goes up from about 400,000 in the simulated data set to 600,000, but the number of reads is 30 million, right? So it's a marginal difference with respect to the number of reads. So it's still computationally efficient. It translates into a few seconds per sample in terms of wall clock time inference. And then, so here we're sort of showing, as we increase the number of reads, how the time scales. And so here it's sort of difficult to see because of the relative sizes of the bars, but this approach is essentially exactly as fast as the approach that doesn't use the improved factorization. So the only other question is, what do we lose in terms of memory? And so here you could imagine you might have to keep more information because you're keeping a more refined representation of your data. But here we sort of show the red bar is salmon, RF, the orange bar is the original salmon, the blue bar is RSEM. And so as you increase the number of reads, RSEM scales linearly with it because it has to remember these values for every fragment. The memory usage of salmon and salmon RF are approximately constant, and the relative difference is very small. So we're not paying more in memory either. Express clearly wins here in terms of absolute memory usage. It's a streaming algorithm, and so it actually doesn't keep these things stored in memory. It updates and does an EM online. So in conclusion, I'd like to, sort of tell you what we took away from this is that the standard approach works well, but it does leave something on the table, and that data-driven factorizations, I'm not saying this is the best one or the only one, but you can recover that lost modeling fidelity without compromising memory or speed. And so there's some interesting approaches or interesting things that we're looking at in future work. So we explored here how well it affects maximum likelihood estimates. But a trend that's been around for a while, MM-Seq does this, and now Callisto and Salmon do this, is estimating variance. And so we're interested in seeing how this affects the estimate of the posterior variance. We're interested in looking at how such factorizations could affect things like allele-specific expression, and also how it interacts with different optimization methods. So here everything was the EM method, but there are other methods that have been shown to work well for RNA-Seq, like variational Bayesian EM and the variational Bayesian natural gradient approaches. So we're interested in looking at those combinations. So in conclusion, I'd like to thank all of the members of my lab. The students are highlighted here in bold if they were authors of this paper. I only have one student who's not on Twitter, so you can't harass her on Twitter to get on Twitter, but I highly encourage you to harass my other students to get her on Twitter. And all of these are implemented and available on GitHub.
Thanks, Rob. There's time for a few questions. Okay, I guess we're getting closer to lunch. Okay, so no specific question. Well, I guess we saved some time. I'm sorry.
Right, sure.
Thanks so much. Huh?
Thanks.